This is a tutorial for BioLitMine. I'll be showing how to find relationships between genes, mesh terms, pathways, and people from PubMed literature. So first, I'm going to show the gene to mesh word cloud search. So I'm going to use wingless for this example, and for the filters, I'm just going to use all results. So this first page, you're going to see the query information, which shows the gene that was mapped from your search term, and then a table of orthologs for the gene you searched. On the left, there's options to filter the orthologs. By default, it's only showing the best match, and you can view the results for the gene you searched or any of these orthologs. We'll just go with the gene we searched for now. So this is showing you the word cloud, and it's for the anatomy mesh category only by default. Since I used wingless, it makes sense that wings would show up the largest. And then down here, there's also a word cloud of the mesh categories. Right now, there's only one, so that's all we're seeing there. And then to update this for a different category, you can just select that here, then click the search again to update. Now it's showing it for the other category. When you have a word cloud you like, you can then export it either to a PNG or SVG. There's a link for exporting the mesh word cloud as well as the category word cloud. And then to view these results as a table, you can click here or choose the gene to mesh option when searching initially. This is going to show you table of the mesh terms associated with the gene that was searched, ordered by the number of publications, and there are link outs for each of these to the actual publications. Going back now to the home page, also going to show a quick example of how the gene lookup works. So I mentioned that it's going to show you the gene that your search term is mapped to. This also works for synonyms, so if you search nub, it shows nubbin as expected. But if you enter a synonym, for example, like this, it's again going to still map it to the same gene. All right, next I'm going to be showing the gene to gene search. So this is going to identify co-citated genes. Just use wingless again. And again, with all these searches, it shows you the orthologs, so you can alternatively view those results, but we'll just go with our search term. Let this load for a minute. While it's loading, I'll point out that the results can be filtered by species over here. And you can search for additional genes here. Alright, so now we have the results for the co-citated genes, again ordered by the number of publications with link outs for each of these. So next I'm going to show the graph view for this co-citated gene search. So this is now showing the gene that was searched in the middle as well as co-cited genes around the edges. And the edges from the search gene represent different types of associations. So there's protein-protein interactions, interlogs, genetic interactions, genetic interlogs, and of course publication associations. Okay, next I'll show the mesh to gene search. So before I do this, I want to also point out that at the bottom there are some useful links for finding mesh terms and for mesh tree views. In this case, we'll search for the stem cells mesh term. And this first page shows the child terms that were found from your search, as well as the mesh term that was mapped to from your search term. And then before we see the results, we're given the option to include the child terms, but this will slow down the search. So for now, we're just going to only use the search term. Alright, so these are all the genes that were associated with that mesh term. Again, organized by publication count with all kinds of link outs here. So with these results, you can then export them with this button, or if you prefer, you can remap to their orthologs. Before I do that, I'm going to filter these so the ortholog mapping isn't too slow. Let's go with top 10 results only. It's going to ask us again if we want to include the child terms. Now we have just the top 10, so I'll remap to the orthologs. I'm going to use a pretty high cutoff just to do this quickly, but I'm going to map from fly to human. Click here, so now it's showing the initial results that were found from fly here, as well as the orthologs, the human orthologs in this side of the table. And again, this can be exported from here. Okay, so next is the gene to people search. So this is going to be useful for identifying PIs that are working on a particular gene of interest, and shows the count of publications that each PI has for that particular gene, as well as the most recent publication. This can be exported here, and again, there are filters here. Alright, so next is the pathway search. So first we'll do pathway to genes. This example, let's use the circadian clock. So these are showing all of the genes that were associated with this pathway, and then the publication count in this column. And again, this links out to the publications. To do another search without going back to the home page, you can just select a new pathway here and click search again. Now, pathway to people. This one works similar to gene to people, only using a pathway. And again, table is in the same format, so I won't go over that again. Alright, so now I'm going to show the enrichment analysis. So we can identify mesh terms that are enriched in a particular gene set. For this example, I'm going to use an autophagy gene list. Let's copy these quick. Alright, so as you would expect, the most enriched mesh term is autophagy. But there's also some other results, and it gives you the count of genes for this mesh category, or this mesh term rather, the number of genes from the input gene list, the score, in this case lower is better, and then the p-value. And over here it'll also show you if any of the gene IDs you searched were not found, which can be useful. Alright, so last I'll show the batch search for publication counts, and this is going to be useful for identifying well-studied or less well-studied genes. This example I'll use kinase genes, and these are going to be generally well-studied, but even amongst a well-studied set of genes, you'll still be able to see that there are some less studied. So we'll click that. There's quite a few genes in this list, so you can't read it quite at the bottom, but if you hold your cursor you can see which genes are showing up at which end of the graph, and then of course we have a table view, and you can sort this by paper counts by clicking on this column, and again linking out to the actual publications. So you can see top gene as well as some less studied. And that covers the basics. If you still have questions, our contact information is here, and our bug report form is here. Thanks for watching.